You're all experts now at the structure of DNA, and so tonight what we're talking about is DNA replication, so how DNA makes more copies. So why does DNA have to replicate? Well, you remember from when we talked about the cell cycle and mitosis and meiosis, DNA has to replicate before the cell can divide. And so remember that replication is one of the things that happens during interphase of the cell cycle. So during interphase, the cell grows, replicates its DNA, grows a little more, and then the cell can divide. And here's what happens during replication. So we start out, remember from our activity in class today, we start out with a double helix, right? So you see this blue double helix over here? And what we have to do first is unzip that double helix. And it's an enzyme called helicase that unzips the double helix. Remember, enzymes are going to end in ASE, so that tells us that this is an enzyme. And then enzymes are often named for what they do. So you think of helix, helicase, it's going to unwind or unzip that double helix. And then once the double helix is unzipped, see how it's unzipped here? Then there's free nucleotides just hanging out in the cell, and they're going to start matching up with their complementary bases. And there's an enzyme that helps with that also, and that enzyme is called DNA polymerase. So DNA polymerase helps the complementary base pairs on the free nucleotides that are just hanging out in the cell, helps them match up on each side of the double helix. So you see it's happening on both sides here. And then it's hydrogen bonds that are going to bond the complementary bases in the middle of the two new double helices. So the line that goes in between the A and T and the line that goes in between the G and the C, those are hydrogen bonds. A couple more details I want you to be familiar with. There's two different strands of the double helix, right? So once the double helix opens, one of the strands is able to be replicated in the direction that the double helix unzips. And then the other one gets replicated in the opposite direction. So the two strands are replicated in opposite directions. The one that's replicated in the direction that the double helix is unzipped is called the leading strand. The one that gets replicated the opposite direction is called the lagging strand. And because of that, because we have two different directions of replication on the two opposite sides of the double helix, we call the replication in opposite directions, we call it anti-parallel. Because the two sides of the ladder are parallel to each other, but they're getting replicated in opposite directions, so anti-parallel. And then the other important thing is that when DNA is replicated, so look over at the image here where you start out, this original parent strand of DNA is all blue, right? But then we're going to unzip it, and half of it over here, half over here, and then the pink are the new nucleotides that are going to form the other side of the double helix. So you're going to result with half old and half new for each of the new strands. They're going to be half old and half new. And the word to describe that is semi-conservative. So conservative would kind of mean keeping the old, right? If you're conserving what you have, is keeping what you have. And it's semi-conservative because you're keeping half of what you have, but half of it's new. So this is where the standard notes are going to stop. And so for those of you doing standard, there's some practice stuff on the other side of your sheet to work on now. Those of you in honors, more details we need to go over. So DNA gets built or put together from the 5' end to the 3' end. So on this image here, you see that on the leading strand, since DNA always needs to be built 5' to 3', that leading strand template is 3' to 5'. See, that's a 3' right there. So on that leading strand, DNA replication can happen continuously. The nucleotides just match up, and it goes straight down the line and keeps going as the DNA unzips. But on the lagging strand, since the template is 5' to 3', you can't just start building in the direction of the unzipping. So instead, the DNA has to be made in these little fragments that get replicated backwards. So they get replicated 5' to 3', 5' to 3', 5' to 3', like that. As the DNA unzips, little fragments are made just a little bit at a time. Those fragments are called Okazaki fragments, named for the person that discovered them. And then all those Okazaki fragments end up getting joined together by another enzyme called DNA ligase. So as those little fragments get made opposite the direction of unzipping, eventually they all get sealed together by DNA ligase. So for you, also for honors on the back there's some extra stuff to work on to make sure you understand all these concepts. And we'll get some more practice in this week in class. Have a great night.